This is genetics lecture number 3. Did you catch the mistake on lecture number 2? Hope you did. Okay, let's do a quick recap of what we saw there. In that lecture, we were introducing the 4 basic crosses of Mendelian genetics. So what makes it Mendelian genetics is that there's one gene. Each gene has 2 alleles, and one allele is dominant, and the other allele is recessive. So one allele is dominant and the other one is recessive. All right, and because of that, there are 4 basic crosses to know. First cross, homozygous cross with homozygous. And it doesn't matter what kind of homozygous we're talking about, dominant or recessive. If you cross a homozygous with a homozygous, your genotype ratio is going to be 1 to 0, and your phenotype ratio is also 1 to 0. If you were to cross a homozygous dominant with a hetero, your genotype ratio is going to be 1 to 1, and your phenotype ratio is 1 to 0. That was a mistake. All right, and number 3, if you cross a homozygous recessive with a hetero, your genotype ratio is, again, 1 to 1. Your phenotype ratio is also 1 to 1. And finally, in number 4, this is a very easy one. Hetero cross with hetero. Your genotype ratio is 1 to 2 to 1. And your phenotype ratio is 3 to 1. All right, so that's a review of lecture number 2. Those are the 4 basic crosses in Mendelian genetics where you have one gene and each gene has 2 alleles. One of the alleles is dominant and the other one is recessive. You can always expect to get these results from those 4 crosses. Okay, let's move on to other vocabulary. What does it mean when we say locus? That means location. And you might see that on exam number 3. So just know that locus is location. And I'm going to show you in the next slide what I mean by that. You also learned, as vocabulary, that to be homozygous means that individuals have the same allele, and to be heterozygous, the individual has 2 different alleles. So just remember that going forward. Here's an example of genes on specific locations on a chromosome. The P's here, for example, are kind of close to the centromere, which is really, really off to the side. Not all chromosomes have a centromere right in the center. This chromosome is in the middle of the P's and the B's. This A gene, I meant to say, is in the middle of the P and the B genes. And the B gene is located at the far end of a chromosome. Those are their locations, those are their loci. Notice that for the P's, they're both the same, big P, so that's homozygous dominant. For the A's, it's lowercase a, so that's homozygous recessive. And then for the B's, you have one of each, a big B and a little b, a dominant and a recessive, so that's a heterozygous. So that's just more practice of the vocabulary that we've been looking at. All right, also, so you don't forget, if a female has big C, little c, the law of segregation separates those. Big C goes there, little c goes there. If a male has big C, little c, they separate those out in the law of segregation also. Big C goes there, little c goes there. And that represents gametes, and the gametes can fuse together to give you offspring, right? That's your Punnett square. That's a good visual of how the crosses work. And now this is a monster cross. What's going on here? Okay, in this cross, we're looking at two genes at the same time. There apparently is an R gene, and then there's a Y gene. And I'm just showing you the dominant alleles. There's also a lowercase r and a lowercase y. But there's a gene that is given the symbols big R's and little r's, and a gene that is given the symbols big Y, little y. And they stand for something like, color and texture. Yellow round, yellow wrinkled, green wrinkled. That's a huge Punnett square. How does this make sense in the world of meiosis so that we can graduate you to understanding this, just like we did with one gene? How does it work with two genes? All right, these two genes happen to be on separate homologous chromosomes, or on separate pairs. It would look like this. On one, you would have like big R, little r, and on the other one, you'd have like big Y, little y. And if you did the loss segregation, you'd find this. These two would be inherited together, and you'd get big R with big Y. These two would be inherited together, and you'd get little r, little y. But did you remember that in meiosis, there's a law that says that this chromosome pair doesn't care what this chromosome pair is doing. So they could have lined up like this also. You could have had, again, the big R, little r in the same spot, but the Y's could have been in a different spot. And in that case, if the loss segregation takes over, you'd have big R with little y. See, they're different now. Or you can have little r with big Y. So that's different from this outcome. So the law of independent assortment takes effect whenever you have genes on separate homologous pairs. Genes on separate homologous pairs. And that gives you 1, 2, 3, 4 different outcomes. 4 different sperm or 4 different eggs. And that's why you would have something like 1, 2, 3, 4 different gametes instead of what we've been seeing, which is like 1, 2 gametes. All right, hope that made sense. You can go back and review it. But this is now going back to meiosis and kind of forcing us to think about the law of independent assortment and how that helps create different gametes. And whenever you look at one gene, then you only have to worry about one homologous pair segregating, giving you 2 gametes. But whenever you keep adding homologous pairs, you're creating more and more and more randomness, and therefore more and more and more gametes. And the Punnett square gets massive. All right, we're never going to do a massive Punnett square. We shouldn't do that, right? Instead, we can do something simpler. Let's look at the laws of probabilities. As an example of how to address a big cross that requires a big Punnett square, how we can do it with a small Punnett squares instead, using a little tiny bit of very easy math. All right, so how about we go with, let's say one parent has big B and little b, and also has another chromosome pair that has, say, big S with little s. And the other parent is also big B, little b on one chromosome pair, and also big S, small s. Wow, those are ugly. Big S, little s on the other chromosome pair. All right, we shouldn't be doing a massive Punnett square like this one. That's just too much. That would be BS. So instead, let's do what's easier. Let's just do 2 small Punnett squares. Let's just do the B's by themselves, off to the side. Big B, little b, cross with big B, little b. All right, we're done. Super easy, super fast. We don't have to do a big old Punnett square. We can also do the S's. Big S, small s. All right, what if we had been asked, what are the chances? What are the chances? See, not ratios, not 1 to 1, 1 to 2 to 1, 3 to 1, 1 to 0. We're not being asked that. We're being asked, what are the chances of getting a child that is born with little b, little s, little s? Well, all we have to do is look in here and look for something that has little b, and there it is. There's one. There's 1 out of 4. So I'm going to put a 1/4 under that. And then we can ask, okay, what are the chances of getting little s from this square? And it looks like there's also 1 out of 4. So I'm going to put a 1/4 right there. The law of probability says all you have to do now is multiply them across. The law of probabilities should also be known as the law of multiplication. And I think that's easier because it reminds you that you need to multiply, right? When do you multiply? Whenever you have multiple genes, like right now. I've got B's and I also have S's. That's multiple genes. Those are two different genes, the B genes and the S genes. So you multiply. And here I am multiplying. When else do you multiply? Whenever you have multiple pregnancies. We're going to see examples of that too. Okay, so let's complete this multiplication. 1 times 1 is 1. 4 times 4 is 16. That's our answer. We're done. And we did not have to do a massive Punnett square to figure that out. Where you have to dig in, you have to fill in all those little squares. That's just too much time. So we instead do 2 tiny Punnett squares. That's faster, less likely to make mistakes, and then you pull out the information that you need. Okay, now you try it. What if the question had been, what are the chances of getting big B, little b, and big S, big S? Okay, here's big B, little b. It looks like there's 2 out of 4. Isn't that 1/2? And what about on the other side for the S's? Looks like there's 1. That's 1 out of 4. So I'm put a 1/4 right there. And I multiply. 1 times 1 is 1. 2 times 4 is 8. Done. Law of probabilities. Now you want to practice the law of probabilities because that's going to be on your extra credit next week. Okay, laws of probabilities are not terrible. Let's do now an example with multiple pregnancies. All right, in this case, let's say we have like big D, little d, cross with big D, little d. All right, we're back to a simple cross. We're not doing the big 2 genes crossing, which is called a dihybrid cross because you're looking at 2 genes, dihybrid. This is monohybrid, which is what we've been doing more of. This is only 1 gene. Okay, so let's say that we do that Punnett square. That's an easy one to fill in. Big D, big D, big D, little d, little d, little d. And let's say that we're asked, let's say that this is like dwarfism, where getting at least 1 big D makes you a dwarf. All right. Or we can do it like a dimple chin versus non-dimple chin. That works too. Let's go with that. You might remember that if we talked about it in lab, a dimple chin is dominant over non-dimple chin. That's a little d. So if you're big D, or big D, little d, you're going to have a dimple chin. If you're little d, you're going to have no dimple on your chin. So here we have 2 parents, both of them heterozygous for dimple chin, and they cross, and we want to know, okay, what are the chances that they will have 3 kids, and all of them will not have a dimple chin? All right, baby number 1, baby number 2, baby number 3. All right, what are the chances that the first child will have no dimple? Looks like there's only 1 out of 4, right? What are the chances that the second child will also not have a dimple? Again, it looks like it's 1 out of 4. All right, what about the third child? What are the chances that it'll not have a dimple? Again, same parent, so it's still 1 out of 4. Aren't these multiple pregnancies? Yes. Multiples. Multiply. 1 times 1 times 1 is 1. 4 times 4 is 16, times 4 is 64. That's the law of probabilities, also known as the law of multiplication. When do you multiply? When you have multiple genes. Dihybrid crosses, for example. Multiple genes, B's and S's, bunch of BS. Or whenever you have multiple pregnancies, multiply. Super easy, straightforward. Can you fish that out of a question? 2 parents with this gene and these genotypes want to know what are the chances that they will have a child, 3 children or 2 children or 4 children with this particular phenotype or genotype. You just do the 1 Punnett square or 2 Punnett squares and then multiply across. Let's keep the same parents and then you try one. What if you were told that these parents want to know what are the chances that the 3 children they plan to have will also inherit the heterozygous condition that they have? Heterozygous. Aren't these the heterozygous children? Aren't there 2 out of 4? So it would be 1/2 in the first pregnancy, 1/2 in the second, 1/2 in the third. Multiply across. 1 times 1 times 1 is 1. 2 times 2 is 4, times 2 is 8. Done. This isn't terrible math, but it's math wrapped into the concepts of genetics and also a tiny bit of meiosis. And so that could potentially make it a little bit more challenging. Okay, let's try this again. This time, let's say we have like, what would be good? Let's go with like big R, little r, big T, little t, cross with big R, little r, also big T, little t. I'm just using a lot of heterozygous examples here, but any one of them could have been homozygous dominant or recessive. The idea is you would just do a Punnett square. All right, so let's say that in this case we want to know what are the chances that they're going to have this child, little r, big T, little t, in the first pregnancy, and that in the second pregnancy, what are the chances that they'll have this, little r, big T, big T. That's what they want to know. What are the chances? Okay, so whenever you're asked for chances, probabilities, odds, likelihood, that's when you're going to give an answer as a fraction. These are not ratios anymore. It's not 1 to 0, 1 to 1, 3 to 1, 1 to 2 to 1. We're not doing that anymore. The only time you do ratios is when you're explicitly asked, what are the ratios? Do a ratio. But if you're not, you're being asked. Odds, likelihood, probabilities. Okay, so what are you supposed to do here? Well, remember, you don't want to do a massive Punnett square, so let's just do the r's first, right? Big Punnett square. Big R, little r. Big R, little r. And then fill it in. And then do the t's. Okay, let's go down here. What are the chances of getting little r, little r? It looks like 1 out of 4. What are the chances of getting big T, little t? Looks like that's 1/2. So the answer here is 1/8. Okay, let's go to the second child. What are the chances of getting little r? Well, look again, looks like it's the same, 1/4. What about big T, big T? Looks like that's also 1 out of 4. What are the chances of this happening? 1 in 16. All right, do you see what we got to do next? We just did the multiple gene part, right? R's and T's. Now we got to do what? Multiple pregnancies. You got 1/8 times 1/16. So for the multiple pregnancies, you multiply those now. 1/8 times 1/16. Let's just leave it there. Whatever the answer is, doesn't matter. Did you figure out that that's what you needed to do next as your next step? You're done. I don't care about the answer. I care about the concept that led to the setting up that potential answer there. That's what matters. That's the law of probabilities, law of multiplication. And it was combined. You had to do it one time for one offspring. You had to do it a second time for the second offspring. And then again, you needed to multiply the first child against the second child to get that formula. The answer doesn't matter. Did you see that that was the logic to go with? Okay, that's the law of probabilities. That's going to be your extra credit next week. All right, study.